what works, what doesn't. It's a tough problem.
And with Alzheimer's, we're talking about cancer, but do you have what are the prospects we're seeing for Alzheimer's?
I don't want to make any generalizations there. I mean, it's really tough. You know.
Okay, no, it's because of the neuroscience that you're working with.
Yeah, I think eventually, we don't know what causes it right now. That's the trouble.
Okay.
And we know some of the molecules involved, and that's because of genetics. But the initial trial, so there's a molecule called beta amyloid, which accumulates in the brains of patients with Alzheimer's. And so there was a hypothesis the whole disease is due to the accumulation of that stuff, and if you could prevent the accumulation, you prevent the disease. And it's not clear whether that's the case. So there's been antibodies developed to that, which look like in animals they decrease the abundance of the stuff in the brain. Whether that's going to work in people is presently unclear. There's some positive indications and some not so positive. It's hard. The other part of the other challenge, of course, is the brain's inaccessible, in a way that blood isn't, and so that makes it much more difficult. I'm optimistic in the long run that we'll get insight whether...
Well, I'm pleased that you're optimistic because that relates to my next question. I mean,
I'm always optimistic. To be a scientist, you have to be pathologically optimistic.
Okay. I mean, let's say we could go 30 years in advance. Do you have any vision of what will the situation be? What are the most exciting possibilities, let's put it that way?
I think any scientist any sensible scientist wouldn't dare make that prediction, because if you look back 30 years ago, the most brilliant person, Watson and Crick, more than 30 years ago, wouldn't have begun to imagine where we are now.
Exactly.
It's based on their discovery. Right. So that's one reason I'm optimistic, is that new stuff comes along and it's simply incredible. Right. And medicine's not going to get cheaper, but it's going to get better.
Good.
Don't tell Stephen Harper I said that.
Okay, I have just a final question. With our mastery of the human genome, how will this transform us as human beings?
I don't think. There's all kinds of, I think, understandable concern about people, looking to have only children who have an IQ 150, and this is rubbish because you can't predict. There's no set of genes which are going to tell you whether your child's going to be good at this or good at that. There might be a specific gene which makes it likely that your child is going to be able to run faster than another child, this kind of stuff. But it's only a likelihood. With 23,000 genes interacting with each other and with the environment, and with a mixture of a set of such genes from your mother and from your father, the predictive abilities are very poor. And so you can get your DNA now sequenced by companies in the US. One of them is called 23andMe. 23 is the number of chromosomes. And they'll give you some predictive testing, which is frankly, it's irresponsible. And it's hilariously inaccurate in some instances. So there's one scientist who had his DNA sequenced, and they told him that, I can't remember what it was, that he was unlikely to ever have a heart attack or something like this. And of course, he'd had a heart attack 4 years before. I mean, this is naive and irresponsible, and it's driven by desire to make money rather than responsibility. Now, on the other hand, these companies, not to badmouth them, they can serve, and 23andMe actually is a good citizen in this respect. They're contributing the DNA sequences and information they get to the general database, so it helps develop a better genetic portrait of humanity in general.
Okay, well, thank you very much for sharing these questions, these answers with us.
Good questions.
Now I'd like to invite the audience to raise their questions.
Do I have to answer them?
That's up to you.
Thank you.
Just regarding like 23andMe, is there a point now where they relate genetic diseases to specific haplotypes? Like, is there more association between,
Well, without going into what a haplotype is for most of the audience, it's just not needed anymore because you can look at individual genes. You know, the cost of sequencing a whole genome is going to be about $100 within a couple of years.
You mean for one person?
Yeah. Yeah. So, you know, all those indirect measures by looking at haplotypes and other things will be largely unnecessary.
but are there any patterns that you're seeing, like, you know, I think originally there was like an African deficiency, I can't remember what it was.
Sickle cell disease.
Sickle cell.
And then there was a Jewish thing, and, you know, so there are specific patterns, like, do you see those developing?
Those, what you're referring to is just a way of looking at the history of genes by looking at the environment in the chromosome in which they were located. And now it's just a waste of time. You can look at the actual gene itself. So that's, historically, that was invaluable. Currently, it's archaic. All within the period of 30 years. Did you spend a lot of your life working on haplotypes? Is that
You mentioned that you had 23,000 genes interacting. So I'm wondering how you go about determining the effects of one single gene when there's so many others.
That's a smart question. If you want to come and work in my lab, let me know. What you're referring to is how do we understand how all these genes interact with each other and talk to each other, even within a cell. You know, in a single cell, not all 23,000 genes are active. In general, about 8 or 10,000 are. But still, that's a lot. And then the question is, how do the cells interact with each other? So this whole area of research is called systems biology. How does the system work? And it's a terribly difficult problem. There's some wonderful scientists, including many wonderful Canadian ones, working on this. And it's so one of the areas is called signaling. How do the cells signal to each other? How do they signal to each other between the nucleus and the cytoplasm? It's very complicated. It's going to take decades to work it out. That's the tough answer. It's a very good question. But that's why, you know, affecting one single gene often has no effect whatsoever, except in these extreme situations that I mentioned, these 6,000 single gene diseases.
I was just wondering if a patient has a liver or lung or heart transplant, is there any genetic factor that would follow, do you think?
As a result of getting the transplant?
Yeah.
No. Because the heart has its own set of genes, but they're not going to influence the genes in the rest of the body, so, which are the recipient's genes.
Doesn't change the personality of the person or anything like that. There has been mention of that made
No.
in the weirdo stuff.
Hi. I was interested in your descriptions of how breakthroughs take place in research, and in particular, I was looking at listening with interest in some of the ways you talked about how you led to breakthroughs. For example, you said the most important thing was to find the right question, and that you had to spend a lot of time working on finding that question and asking it at the right time. You also talked about going where the research takes you. You talked about how 20 years ago, when the real breakthroughs were coming about, that it was important to be a good scientist, you had to be imaginative. Whereas now you can be a good scientist and be a problem solver.
Yeah.
All of those ideas of questioning, of going where the research takes you, of being imaginative, made me think of two other words, which would be creativity and intuition. And I just wanted to ask you how much you thought to being a pioneer in science, how important it is to follow your own intuition and to be creative as part of that process. And does, when you were talking about the culture of great institutions, whether they're universities or great research hospitals, does that culture contribute to the creativity that the scientists have?
Oh, completely. I mean, because, in a place you're encouraged to ask great questions and challenged to address them and stimulated to have the chutzpah to take them on. That's one of the major differences. The belief that you can actually tackle tough questions and solve them is an inherent characteristic of great institutions. You've made a lot of wonderful points. I don't disagree with any of them. I mean, if you're a great problem solver and creative and imaginative and intuitive, you're going to win the Nobel Prize more likely than the girl next door. So, yeah, all those things are important. But now it's just what I meant was, there are some scientists who are fundamentally problem solvers and others who are they're successful because they've got great imaginations. They might go to the guy next door and say, I've got this problem, and the guy next door is a problem solver. How do you think I should address it? The two things are not mutually exclusive. They're very complementary. And yeah.
Thank you. I've talked long enough.
Okay.
Do you find out that scientists work alone and try to prove their own initiative, or are they team workers?
No, so that's a wonderful. The question is, do scientists work alone, or are they team workers? And the question is very timely. The culture of biomedical research is changing completely. So you may know that physicists, the major papers in physics now have 100, 200, 300 authors. And institutions now give credit if you're one of 300 authors on a major paper, because that's the only way it can occur. And that's what's happened in biology and in biomedicine. It's too complicated to expect that any great story is going to likely emerge. There are still many papers with a few authors, but the numbers of biomedical papers with 15, 20, 30, 100 authors is increasing all the time. And that's because it's tough to do. And you need many people contributing many different perspectives and areas of expertise. So the NETO-1 research project that we discussed earlier involved 3 labs working for 8 years and had about 16 authors. It's not easy.
Do you appreciate that? Do you find that is a good working situation for scientists? Or do you think that the individual is still the imaginative and creative scientist?
I think both are, again, they're not mutually exclusive. You get 3 different, so the 3 labs that contributed this paper were John Roders at Mount Sinai Hospital in Toronto, my lab, and Mike Salter, who was a neurophysiologist at Sick Kids. And John Roder was a mouse learning expert. So the 3 of us worked together very well. When you're looking for collaborators, you want people who are really smart, that are going to add something, and that are decent people to work with. The last is a really important criterion. Like, there's some people who, if they were the last scientists on Earth, I'd never collaborate with again.
You mentioned that the genes talk and sing to each other. Do you find that the scientists in different fields are doing that?
In biology, you can get any 2 biological researchers, doesn't matter whether they're in medicine or studying plants. They can sit down and they're talking the common language. They use the same kind of tools. It's all one field now. It's, the divisions of biochemists and cell biologists and geneticists are totally artificial. They exist for the purposes of teaching. But, in my lab, we do genetics, biochemistry, cell biology. That's the same stuff that any biomedical researcher in a lab around the world does. And it's a common language. It's kind of fun, actually.
Any others?
To you. Is there a dilemma for a scientist? I mean, her question kind of leads into this, between...
No, it's working.
It's working.
It's working.
Is there a dilemma for a scientist deciding between sharing his or her information with other scientists or holding out for recognition and, or more importantly, big pharma money?
Yeah, the money isn't a big part of it. The money part of it is getting research grants to do more research. I mean, if you've got a story, you want to be the one with your colleagues working on it with you to get it out first, to get the credit for it. If in this NETO paper we talked about with 3 labs for 8 years, you know, after 3 labs in 8 years, and I wouldn't want to add up the total budget for that paper, it's probably $2 million. You want to be there first, you know, to pick up a journal and realize